Welcome to the deep dive. Today, we're plunging headfirst into viral adaptation, a really critical topic if you're stepping into the medical field. Think of this as your shortcut to understanding why viruses are these ultimate master chameleons, constantly shape-shifting in this ongoing evolutionary arms race with us, their hosts.
Absolutely. And for aspiring medical professionals like you, grasping viral evolution isn't just about, passing an exam. It directly shapes how we approach patient care, how we develop life-saving vaccines, and strategize for public health crises. Our mission today, to break down these complex adaptation mechanisms, we'll use some memorable anchors, vivid metaphors, and maybe pose a few thought-provoking questions, all designed specifically for your retention and recall.
Exactly. So let's kick off with the big question driving all this. What makes these microscopic entities such formidable shapeshifters? How are they constantly outsmarting our natural defenses and even our most advanced medical interventions? Okay, let's maybe unpack this a bit. Before we dive into how they adapt, what exactly is a virus at its core? For those of you knee-deep in virology, you know the basics, but let's quickly underscore what makes them so unique and, frankly, relentless.
Right. At its most fundamental level, a virus is, well, it's an ingenious minimalist entity. Reliable medical sources tell us their genomes can be RNA or DNA-based, single-stranded, double-stranded, lots of variety there. But what's often overlooked, I think even by seasoned students, is the sheer elegance of their obligate intracellular parasitism. They
Right.
completely lack functional translation machinery. They have no way to make their own proteins, which means they're entirely dependent on hijacking a host factory to replicate and spread as viral particles or virions.
Totally dependent.
Totally. And what's truly fascinating over evolutionary time is their ability to actually integrate into cellular genomes. They've massively enriched the genetic repository of countless organisms over eons. Their overarching mission, if you want to call it that, is incredibly clear. Adapt. Adapt to their hosts, evade defense mechanisms, take over cellular metabolism for their own benefit. It's all about transmitting and persisting by fixing beneficial mutations that allow for better exploitation of new hosts.
So it's this constant evolutionary pressure cooker.
Exactly. It's a relentless chess match. And every viral move reshapes not just the host's biology, but also our therapeutic and preventive strategies. It forces us to constantly innovate. And this relentless mission, this drive to adapt, brings us to the core biological engines.
Yeah.
Genetic changes, primarily through mutation and recombination. These processes are absolutely essential for generating the new genetic diversity they need to survive and, keep going.
Okay, so this is where the adaptation really comes into sharp focus, right? When we talk about how viruses change their appearance to evade our defenses, we're often talking about two key strategies: antigenic drift and antigenic shift.
Indeed. And let's use some metaphors here to make these really stick for recall. Think of antigenic drift as the slow burn, or maybe a minor costume change.
Okay, I like that.
It's a gradual process. Small, subtle mutations accumulate over time in the gene's coding for those surface proteins like haemagglutinin, HA,
Yeah.
neuraminidase, NA, within a single strain of virus. It's like making small alterations to a familiar outfit, a slightly different hat, maybe different glasses. The overall identity is still recognizable, but there are enough minor tweaks to potentially slip past an immune system that remembers the old look, but isn't looking that closely. It's constant low-level evolution.
Right, the slow burn.
Then you have antigenic shift. This is the big one. Think the major wardrobe overhaul or even viral identity theft. This is sudden. It's dramatic. It happens when two or more different strains of a virus infect the same host cell at the same time. What happens inside is a reassortment of genetic material, basically, a gene-swapping party. And this leads to the emergence of a completely new subtype with a totally novel antigenic profile. It's not just a minor tweak. The whole identity changes overnight. The virus becomes almost unrecognizable to an immune system primed for the old version.
That's a huge difference. Drift versus shift. So thinking about drift, those subtle changes. Is it possible for a virus to gain significant functional advantages through drift that aren't immediately obvious just by looking at the surface proteins? But for you, our listener, let's pause on this. If a virus changes its costume bit by bit through drift versus getting a whole new identity through shift, which one do you think poses the bigger, more unpredictable challenge for our immune system and for vaccine development, and why? Keep that in mind. We'll circle back to the answer shortly. Okay, building on that crucial distinction between drift and shift, let's apply it directly. Let's look at a classic, highly relevant example. Influenza, the flu. This virus is really a masterclass in adaptation.
Oh, absolutely. Influenza is a prime example, and it really helps illustrate the answer to your previous question. It's an orthomyxovirus. That's a specific type of RNA virus. It has an envelope, and critically, a segmented single-stranded RNA genome. That segmented part is key.
Why segment?
Because unlike a continuous strand of genetic material, these segments allow for a high rate of mutation, yes, but more importantly, they facilitate that genetic reassortment, the gene swapping that drives antigenic shift. The pieces can be easily shuffled. Now, for its structure, let's use two key surface glycoproteins as our anchors for understanding. Haemagglutinin, HA, and neuraminidase.
HA and NA.
Got it. Think of HA as the key to the host cell. It's responsible for viral attachment, binding to the cell, and membrane fusion. It lets the virus in. NA, on the other hand, is the escape artist, or maybe enzyme scissors.
Enzyme scissors.
Yeah, it hydrolyzes mucus. It digests sialic acid receptors on cell surfaces. This makes it easier for the newly formed virus particles to get away from the infected cell and spread. We also classify influenza into three main serotypes, A, B, and C. These are distinguished by their internal nucleoprotein antigens, not the surface ones.
Okay, so back to our analogy then. Costume change versus identity theft. Which type of influenza virus is the main culprit behind those really dramatic, unpredictable outbreaks, even pandemics? And what's the key difference that lets it do that?
Right. And if you've thought about the scale of something like a pandemic, the answer really jumps out. It's antigenic shift, without a doubt. And this occurs only in influenza A viruses.
Only in A?
Only in A. This is the identity theft scenario, the major wardrobe overhaul, and it poses the bigger, more unpredictable challenge because it creates wholly new viral subtypes our immune systems haven't seen. The reason influenza A can do this is its broad host range. These viruses infect birds, pigs, horses, humans, many species. Fowls are major reservoirs. This wide range makes it much easier for different strains of influenza A to infect the same host cell, maybe in a pig or a bird. That co-infection is the prerequisite for the gene-swapping party, the genetic reassortment that causes antigenic shift.
So the mixing happens because different strains can meet in the same animal host.
Exactly. Influenza B, on the other hand, primarily infects only humans. This really limits its potential for that dramatic gene swapping. You don't get the same mixing pot. And that's why influenza A causes these sporadic pandemics, often with much higher mortality rates. Whereas influenza B tends to cause more predictable seasonal epidemics, mainly driven by the slower antigenic drift.
Makes sense. And we hear about specific influenza A subtypes. Right?
Yes. Common human ones include H1N1, H2N2, H3N2. These are formed from combinations of currently 16 known HA subtypes and 9 known NA subtypes found in nature.
Wow, lots of potential combinations. Okay, so the virus changes its identity, particularly influenza A. Now, what actually happens when this shapeshifter gets inside us? What are the consequences of its attack?
Well, once inside, influenza typically heads for the respiratory epithelium. It prefers to multiply in the ciliated cells of the lower respiratory tract. This causes pretty significant functional and structural damage. It shuts down the cell's own synthesis processes and leads to the shedding of those crucial ciliated cells and mucus-producing cells that normally protect our airways.
So it damages our defenses.
Precisely. This damage messes up the body's natural clearance mechanisms, like the mucociliary escalator, and causes localized inflammation. Interestingly, viremia, the virus getting into the bloodstream, is actually quite rare with influenza. It tends to stay localized in the respiratory tract. Now, here's another question for you, our listener, to think about. Knowing how influenza damages that delicate protective lining of our airways, what kind of secondary invaders might take advantage of this situation? And why would that be particularly dangerous, especially for vulnerable patients? Think about what normally keeps bacteria out. The common signs and symptoms, most people know these. Sudden fever, runny nose, sore throat, congestion, cough, muscle aches, myalgia. People feel utterly wiped out, sometimes vomiting and diarrhea too. But the real danger often lies in the complications, things like acute bronchitis, pneumonia, and it can really exacerbate underlying chronic respiratory diseases like asthma or COPD.
And those secondary invaders you mentioned.
Right. To answer that question, the damage the virus does to the lung epithelium creates literal gaps in our protective barrier. These gaps make the host highly susceptible to secondary bacterial infections. Bacteria that might normally be cleared away can now invade. The three most common bacterial culprits causing post-influenza pneumonia are Streptococcus pneumonia, Staphylococcus aureus, and Haemophilus influenza.
Haemophilus influenza. That sounds like it causes influenza.
Good catch. It's a bit of a historical misnomer. It was named Haemophilus influenza because it was frequently found in patients during flu pandemics, particularly the 1918 one. People initially thought it caused the flu, but we now know the flu is viral. H-flu, the bacterium, is an opportunistic secondary invader.
Got it. And speaking of 1918, that really highlights the power of antigenic shift, doesn't it?
It's the starkest example. The 1918 Spanish flu was an H1N1 virus, but it had genes of avian origin, a major antigenic shift event. It infected, estimates say, about 1/3 of the entire world's population and caused at least 50 million deaths globally. A truly catastrophic, major wardrobe overhaul.
Unbelievable impact. So influenza is a powerful example. But as we know all too well recently, it's not the only viral family keeping us on our toes. Let's pivot to another master adapter, one that's been very prominent lately, the coronaviruses.
Yes, coronaviruses. These are enveloped viruses, and they have a positive sense RNA genome. That positive sense part is significant because it means their RNA can act directly as messenger RNA in the host cell. The host ribosomes can translate it immediately, like it's a ready-to-go instruction manual for making more viruses.
Efficient.
Very. They also have a nucleocapsid with helical symmetry. They primarily infect the upper respiratory and gastrointestinal tracts of mammals and birds. And actually, they're responsible for a good chunk, maybe 15–30% of common colds in human adults. But as the world learned dramatically, some coronaviruses can cause much more severe disease. We have MERS-CoV and, of course, SARS-CoV-2, the virus behind COVID-19. These stand in stark contrast to 4 other human coronaviruses usually associated with just mild cold symptoms.
And with SARS-CoV-2, so much focus has been on its spike protein, that iconic clove-like structure. How critical is that specific protein to its adaptive success and its ability to infect us?
Oh, it's absolutely pivotal. The spike S protein is the coronavirus's signature feature, visually appearing as these clove-like structures, or you could call it a clove-shaped key sticking out from the virus surface. It plays a central role in pretty much everything: pathogenesis, evolution, transmission. This protein is the unit that recognizes and binds to our host cell receptor, ACE2. That binding is the crucial first step, initiating viral entry via fusion.
So it's the lockpicker.
Exactly. And critically, the S protein is also the main target for our neutralizing antibodies, the antibodies that can block infection. Its different subunits in the various shapes or conformational states, it can significantly affect the virus's ability to cause disease, its infectivity, and its evolutionary path, its mutations.
So changes in the spike are central to variants.
Absolutely central. Understanding its structure and how it changes isn't just academic. It's key to predicting how variants might behave and designing effective vaccines and therapies. And we also have to remember the zoonotic origin of SARS-CoV-2, jumping from animals, which again highlights this cross-species adaptability.
That zoonotic leap is fascinating. It brings us to our final ponder question for today. When a virus like SARS-CoV-2 makes that jump from an animal reservoir into humans, it doesn't just instantly cause a global pandemic, right? There's usually a process, a stepwise journey. What are the typical 5 stages of viral conquest a pathogen goes through to become an established, exclusive human disease? How does it adapt along that way?
That's a great question, and there's a really useful framework for understanding this outlined by Wolf and colleagues back in 2007. These 5 stages of zoonotic virus adaptation provide a key anchor. Stage 1. The virus exists only in animals, no human infections yet. It's contained in its natural reservoir.
Okay, stage 1, animals only.
Stage 2. The virus jumps to humans. Spillover happens, but there's no human-to-human transmission. Each case comes directly from an animal source.
Stage 2, animal to human, dead end.
Stage 3. We start to see some human-to-human transmission, but it's rare, maybe just a few cycles. It's limited and usually stutters out without sustained spread.
Stage 3, limited human spread.
Stage 4. The virus achieves established human-to-human transmission. It can spread efficiently within the human population, leading to larger outbreaks. This is a critical tipping point.
Stage 4, sustained human spread.
And finally, stage 5. The pathogen becomes an exclusive human pathogen. It no longer needs the animal reservoir to keep circulating. It's fully adapted to us. This is the end result of all those previous adaptive steps.
Stage 5, humans only. That's a really clear progression.
It is. And understanding these stages, especially with something like SARS-CoV-2, really underscores how vital genome sequencing is. Sequencing helps us track its evolution, understand transmission patterns, figure out pathogenesis, spot immune evasion or potential drug resistance, and ultimately develop effective therapies.
Right. So we've covered a lot about how incredibly adept viruses are at changing. What does this all mean for us? As a medical community, how do we even begin to combat these relentlessly adaptable pathogens?
Well, the significance of understanding this adaptation is profound. It's not just theoretical. It directly impacts how virulent a virus is, how easily it transmits, and crucially, how well our treatments work.
Yeah.
To mitigate this constant adaptation, we rely on three major strategies, drawing from established medical knowledge.
Okay, what are they?
First, vaccination. You could think of this as stimulating the host's sentry system.
Priming the defenses.
Exactly. Vaccination prompts your immune system to produce antibodies and specialized immune cells that recognize and attack the virus if you encounter it later. This prevents the virus from getting a foothold, infecting cells, and replicating. By doing that, it reduces the opportunities for the virus to mutate and adapt within a host. And on a population level, it reduces transmission by lowering the number of susceptible people, which helps limit the emergence and spread of new variants.
Makes sense. Strategy 1, vaccination.
Second, antiviral drugs. We could describe these as interrupting the viral blueprint.
Throwing a wrench in the works.
Pretty much. These drugs work by inhibiting viral replication, stopping the virus from making copies of itself, and thus reducing the viral load in an infected person. However, there's a key challenge. Viruses can adapt against these drugs too. Adaptation can lead to the emergence of drug-resistant strains, which limits the long-term effectiveness of antivirals. Think of siltamivir and zanamivir.
Yeah.
For influenza, they work, but we constantly monitor for resistance.
Right, the virus fights back even against drugs. So strategy 2, antivirals with caveats.
And third, surveillance. This is our early warning system.
Watching and waiting.
More like actively watching and analyzing. It involves the ongoing systematic collection, analysis, and interpretation of health data, monitoring disease patterns, sequencing viral samples. It's absolutely crucial for identifying emerging viral threats as early as possible and understanding their evolutionary trajectories. Where are they changing? How are they spreading? This allows us to be proactive or at least react more quickly and effectively rather than always being caught on the back foot. Continuous vigilance is key.
Okay, vaccination, antivirals, and surveillance, our main toolkit. Wow, that was a truly deep dive into the fascinating and, yeah, sometimes frightening world of viral adaptation. From those minor costume changes of drift to the full identity theft of shift, viruses are just relentlessly evolving. They really keep us on our toes.
They certainly do. We've touched on the fundamental mechanisms, drift and shift. We used influenza as that classic model of adaptation. And we explored that critical 5-stage journey of zoonotic viruses, like coronaviruses, making the leap from animals to become human pathogens. For you, as future clinicians, this knowledge really is foundational for preventing and treating viral diseases effectively.
So wrapping up, what does this all mean for you, our learner, heading into your medical career? Beyond just memorizing the mechanisms like drift and shift, remember that every new viral variant, every evolutionary leap, represents a fresh challenge for public health. It demands your critical thinking, your understanding, and ultimately an adaptable approach to medicine itself.
Absolutely. And maybe here's a final provocative thought for you to mull over, something to explore on your own. Given this constant evolutionary dance between viruses and us, and considering our mitigation strategies like surveillance, if you could design the ultimate global viral detection network to try and predict the next pandemic-level threat, what key pieces of information, beyond just genetic sequencing, would you prioritize collecting and analyzing? What else do we need to truly stay ahead of these relentless adapters?
Something to definitely think about. Thank you for joining us on the deep dive. We hope this conversation helps you navigate the complex world of viral adaptation with a bit more understanding and confidence.